Hello everyone. Today we are doing a topic, tissue processing, and your presenter is Mrs. Celia. We begin by the objectives of the day. The first objective is define tissue processing. Describe all the tissue processing stages. Explain procedure used for selection of tissues for processing. Discuss various characteristics of chemicals and reagents used in the process. So when you go to the introduction, we begin from the definition of terms. Then the first term is tissue. So what is a tissue? A tissue is a group of layer of cells that work together to perform a specific function. Then what is tissue processing? This is the treatment of a tissue with various reagents to enable production of thin sections for microscopic examinations. So to select a tissue, one must consider the following one: the size of the tissue, the agency of the tissue, time for fixation, and nature of the tissues. Under tissue processes, we have the first process being dehydration. So dehydration is the complete removal of water from a tissue specimen. This is done by use of alcohol and acetone. Here we assume a tissue contains water, and therefore this water must be removed from the tissue to facilitate other subsequent processes. So this water is removed by the use of alcohol and acetone. So to begin with alcohol, in alcohol, the tissue is transferred into different concentrations, starting from the lowest to the highest. For example, usually used concentrations are 50%, 60%, 80%, and absolute alcohol. And here in absolute we have three changes. Three changes means the first container having 100% alcohol, the second container also having another 100% alcohol, and the third container also having another 100% alcohol. This takes about 2 to 12 hours depending on the nature of the tissue and the temperature at which the process is being done. Then we go to acetone. Acetone takes about half an hour, and it's very rapid and it's good for agent biopsies. The next process is clearing, and clearing is the removal of alcohol or any other clearing agent from the tissue specimen. And the commonly used clearing agents are xylene, chloroform, toluene, and paraffin oil. So the alcohol used to remove water in dehydration process must also be removed here to facilitate the next process. So here we use the xylene, toluene, or the chloroform. Then the xylene here, we have its advantages, and the first advantage is that it's rapid in action and takes about 2 to 6 hours. Tissues are rendered transparent. And then the disadvantage we have is that it's highly flammable and it can cause excess shrinkage. The third process is infiltration, which is also known as impregnation, and this is the saturation of a tissue specimen with infiltration medium, and it's done to remove the clearing agent and air from the tissue specimen. This gives a tissue an internal support. Remember, here we have a tissue that we've removed water from it, and from removing water we have also removed the alcohol that we used to remove the water. So now we are saturating this tissue with paraffin wax. Why are we doing this? To give it an internal support. The inside must have some support for the subsequent processes to take place. So after giving the tissue an internal support, we also have the next process, that is embedding, which is also known as blocking. And it is a process where a tissue is given an external support by use of paraffin wax to form a mold and facilitate other subsequent processes. So here we've given a tissue an internal support, and now we want to give it the external support. We put that tissue in a mold, and then we put paraffin wax to give it the shape from outside. Then from there we can now cut the tissue into thin sections, where the next process is now microtomy. This is the cutting of a tissue specimen into thin sections using special machines called the microtomes for microscopic examinations. The example of microtome knives include the cryostat, the sliding machines, rotary machine, the freezing machine, and the rocking machines. So under microtome knives, they are used for cutting tissue sections and are very sharp. They are made of steel, diamond, or glass depending on the manufacturer's wish, and are classified according to the way in which they are ground or sharpened, as each is meant for specific function. So the next slide, we have the knife profiles, and if you can check the images, we have the plano concave knife profile, and then the wedge knife profile. And then these microtomes, you must also care about them, take care of them. In taking care of the microtomes, you must ensure that the knives are stored in the boxes when not in use. Knives should never be laid flat on a bench. A knife edge should be sharpened. Never lend or borrow a microtome for misuse. Always ensure that when you borrow a microtome knife, you use it for the right purpose. Then from cutting the tissue into smaller sections, we now go to staining. And staining is now a process of imparting color to tissue section in order to make evident various tissue components. So in this topic, we are going to use hematoxylin and eosin stains. And now the principle of hematoxylin: this is a basic stain and stain the nuclear because it's acidic. Eosin being an acid stain, it is taken up by the basic tissue structure like the cytoplasm. And now the procedure: dip tissue sections in 2 to 3 changes of xylene for about 7 minutes each to remove paraffin wax from the tissue to allow stain penetration. Transfer the tissue to 2 changes of absolute alcohol followed by descending grades. Descending grades mean if it is 60%, the next will be 30%, 20%, and then 10%. So this is used to avoid shrinkage of a tissue specimen when taken to water directly. Then wash in tap water to remove alcohol. Flood the section with the hematoxylin stain. Rinse in tap water to remove the stain. Differentiate in 1% alcohol up to 30 seconds to remove excess stain. Blow in running water for 10 minutes and counter stain in eosin for 3 to 5 minutes to give color contrast to hematoxylin stained structures. Dehydrate in ascending grades of alcohol such as 30%, 50%, 70%, 90%, and absolute to remove the water which will not mix with xylene. Clear in 2 to 3 changes of xylene if it is to be mounted in a hydrocarbon container mountant. Mount in DPX. This is to preserve and protect the tissue section from dust and pest, and also to raise the refractive indices of various tissue structures for ease of identification. Expected results: for the nuclei should be blue to purple. The cytoplasm should stain from red to pink, and the red blood cells should be orange, and the rest of the tissue should have shades of red pink. So images of stained tissue sections of liver and intestines. So if you look at the images, you see they are stained purple, and there are also traces of pink. And then the other images also are for skeletal and smooth muscle. So the first image is for the skeletal muscle, and the second image is for the smooth muscles. So you can just see how eosin and hematoxylin stains and the contrast in the images. And then come to the end of the lesson. Thank you very much.